So we are in a stand, a typical stand that hemlocks would be growing here in Southwest Virginia. About 23 years ago, the Adelgids came through this area, and it wasn't long before they severely impacted the trees. And we've got a stand here, mixed conifers, hemlocks, Virginia pine, white pine, hemlocks in their native, sort of natural setting, riparian environment. We've got a stream right here, and you can see across the way that there's some snags, dead or declining hemlock trees, and as a result, the stream itself has lost the overstory shade, and the temperatures have increased as a result of the loss of the tree. And so that's an impactful in terms of the environment and the aquatic system. So when hemlocks have come, when the Adelgid has come and impacted these trees, we have trees that the foliage is still okay, and other trees where they're just hanging on. And that's pretty much what we've experienced in this part of Virginia, where a percentage of the trees have died and some trees have declined and survived, but at reduced vigor. And then we can see across the way some regeneration coming back, some younger trees with some healthy foliage on it. And so while we're not completely losing the hemlock tree, we have lost many of the attributes that a hemlock forest produced for this site. And in this particular stand surrounding us, typically we're in a riparian area. We've got a stream that is intermittent in the late winter, early spring. It's active. In the fall it'll be dry. There's a lot of rhododendron around here, typical habitat. And in terms of species of trees, we've got a mixed conifers of Virginia, white pine, and hemlocks. And the hemlocks are probably second, who knows, maybe even more than third, maybe a third growth, but they're mature trees and they're in decline. And if you look across here, you can see that there's a lot of snags of dead trees from when the Adelgid first came through 20 years ago and started killing the trees. And what happens is, in the ecological impact, is that hemlocks typically shade streams. And with the loss of that overstory, the temperatures in those streams will rise. And so aquatic organisms are impacted by that. And there isn't really a replacement for the loss of hemlock in terms of providing the kind of coverage that you expected. You can look over there, there's a tree growing out of the side of the edge of the stream, and that's how hemlocks grow a lot of times. And so when you lose, in the case of the hemlocks that have survived, they've lost a lot of their crown, and they're not as vigorous and robust as they were before the Adelgid came in. You can pan, looking over there toward this further distance in the stream, you see some younger hemlocks that are coming back, and the foliage is looking pretty good. So there is regeneration that takes place, but overall this is a heavily impacted site, and I wanted to show you what a typical site would look like, usually along a stream that hemlocks flourish in. So this is one of our older trees at the site, and if you look up and pan up to the crown, you can see that the crown is just pretty much limited to the top of the tree, and the branches, down below the top 80% to down to the ground are basically, there's no foliage. The live crown ratio is really small on this particular tree, and it's an indication of decline. You don't see a lot of vigorous new growth on the tree as well, and so these are indications that it doesn't take much at this point for a tree to get killed off because it's under stress, and so that's typically what is the case. We have a tree around us who that fell over, even though it was alive, but partly because maybe its roots rotted from being under stress and in decline. So we're in a public area, national forest, recreation area, pollinator garden nearby, mixed hardwood, some Virginia pine overstory, and walking through here ran into these patches of understory hemlock, and hemlock that are relatively healthy, hemlock that are infested with hemlock woolly adelgid. And you know, upon searching around and looking around, eventually finding some mature hemlock trees that are providing the seed for this regeneration. We've got one significant tree right over here to the right, and then one in the distance. And so this is an interesting location because these mature trees are pretty healthy, and we're getting regeneration without any help, without any, management, cultural activities to encourage them to grow. So this gives us a lot of hope that, hemlocks, as impacted as they are able to survive in some locations. And we can't really tell you why hemlock is flourishing in this little spot right here, but it is very encouraging for us. You can see that the needles aren't as close here as they were in some of the foliage you saw at the insectary, but nevertheless, almost every needle is being colonized with an adelgid. And this shows you that we've got a reasonable density of the non-native insect species, and because it's on the newest shoots, as the adelgids attack the newest shoots, they then affect the growth of this coming year's shoots. And so with a density this high, this will prevent new growth. And so what happens is what was a healthy shoot will decline, and you will not get additional growth, and it'll look something more like this. So when these adelgids colonize the base of these needles at that high a density, what happens is it prevents the shoot from setting a new bud and growing, and that new shoot won't grow and new needles won't grow. And so then the hemlock is left with only their older needles. And when it's left with only their older needles, then photosynthesis declines dramatically. And that's how the trees start to decline. So without the new shoot growth, conifers cannot survive. And eventually, with the population density of the adelgid so high, it can cause the tree to die. So the hemlocks themselves are keystone species. Usually, when you think about climax species in a succession of a forest, stand over many, many years, usually hemlocks come in a later stage of the successional point of time. These trees are not well adapted to the invasive insect, and they can die very quickly, especially if they're under stress. So we have lost a very high percentage of our old growth hemlock, eastern hemlock, throughout the southern and mid-Atlantic, and now moving up into New England area. Just extensive areas of death and destruction. There are trees that are younger are also impacted, but there is some recovery that we see because the populations of the pests actually fluctuate over time, sometimes due to climatic conditions and other times due to the reduction in availability of host because of the damage that the insects caused. So we do get some regrowth in trees back, but there's been overall this massive decline in mature hemlock health throughout the eastern U.S. So as Carrie mentioned, we put about 200 larvae in here that'll pupate and then change into an adult stage and store them. Now, we may produce 40,000 larvae in a year, but we may only get about 15,000, 20,000 adults emerge. So there's significant mortality that takes place in these containers. And one of the things we're doing for our research is trying to understand better what's going on with these dormant insects in the soil that we don't get a higher percentage of emergence. We have done work in the field to try to assess emergence, and our numbers are actually less in the field than they are in the lab. So it's clear that we don't fully understand what's going on in the soil system. Hi there, everybody. So this is a branch from a eastern hemlock tree, and on this branch you see some white woolly material that's actually a soft-bodied insect underneath it, like an aphid. And these insects come from Asia, and they are now in eastern and Carolina hemlock forests. We first found them in the 1950s. They've been here 70 years, and in the last 30 years they have spread and caused large-scale mortality in the eastern U.S. So it's a very difficult insect to work with, very small, and one of the reasons this insect flourishes so well in eastern North America is the trees themselves are very susceptible, and the natural enemy complex from their native environment isn't present here. So one of the things we do in this insectary laboratory is we rear natural enemies that we found, some in Western North America, where this insect is also native, and also in Japan. And so we've got two versions of the same beetle from different parts of the world, and their goal, our goal, is to produce enough so that when we release them, they will feed on these adelgids and cause populations to decline and keep these trees healthy. So we're going to go through a few steps of actually how we produce large numbers of these beetles and distribute them out to cooperators on federal and state public lands throughout the eastern U.S. So Carrie Jo is going to take you through a few steps right here.
So all of these insects are unique in that they are winter active. So they're primarily out and doing their thing in the environment from approximately October until about May of the following year. In January and February, that's when our production in the lab really ramps up. That's the period of oviposition or egg laying for all of these species. So what we do is we hold the beetles, the Larvae beetles, in quantities of about 15 to 20 in these adult containers. They stay on this foliage for approximately 1 week. And then we go through this foliage every week, pull the beetles off, and then save the branches that they've been feeding on and laying their eggs on. And then we take those to our larval rearing room, where the eggs will hatch and they will develop before larval instarts. The process takes about approximately 6 to 8 weeks before we have mature larvae available.
Show how small the beetles are.
Yeah, the beetles are very small. Kind of the size of a peppercorn is what I usually like to say. But again, we have the two species, Larvae nigrinus, which is native to Western North America, and then Larvae osoptensis, which is native to Japan.
So it's important for you to know that when we first started studying these beetles, we brought them into a quarantine facility here at Virginia Tech, and we studied them for several years to make sure that they only fed on Hemlock woolly adelgid, and that they posed no risk to any other native organisms in the eastern U.S. So because they fed on Hemlock woolly adelgid and no native adelgid species, these were deemed safe for release. And this is a process you go through every time you want to introduce a biological control agent You have to go through rigorous testing and multiple levels of approval before you're given permission to release this biological control agent. First at the federal level and then later at the state level. Keep them in baths of water to keep the needles on the plants, on the cut trees, on the cut branches. So these are foliage that are collected from the forest that have the food that the beetles will feed on. Always on the underside of the branches.
So we take the foliage that the beetles have been on for approximately 1 week. They've been on the foliage, laying eggs, and eating the HWA. We take that foliage and put it into these larval rearing funnels. And the eggs will hatch in approximately 2 weeks, and then they will develop through four instars over a period of 6 to 8 weeks. After that period of time, the mature larvae, their natural tendency in the wild is to drop off of the branches down to the soil surface to burrow in and create a pupation chamber with a pupate. So we intercept this behavior in the lab by using these mason jars. They drop down from the funnels into these jars. We check the jars daily and count all the larvae that have collected here, and then we move the larvae to the soil surface in these soil containers. So approximately 200 larvae will be placed onto the soil surface. The larvae will burrow down, as I said, and create that pupation chamber. They'll pupate for about 6 to 8 weeks, and then they will eclose and become adults. And instead of emerging right away as adults, they stay in the soil and enter into a period of estivation. And then they remain in that estivation throughout the summer, and then in the fall, once the temperatures start to drop and cool down, then they will emerge.
So all these will be filled up by April. The larvae will develop, start collecting daily. Ultimately, these larvae will go into the soil containers. We'll get anywhere from 30 to 50,000 larvae that'll drop over the course of the spring season. Then they go into another room in those soil containers where they'll sit for 3 to 5 months before they emerge, and then we're able to ship them to cooperators for release. So one thing you can't really tell, except for Carrie wearing a jacket, is it's actually very cold in here. It's between 13 and 17 degrees during this developmental season. These are insects, both the adelgids and the predators, that like it cold, and they're active in the winter and they develop in the winter. So we try to simulate the conditions that they would have right now. It's March 9th, and we're trying to simulate those conditions. So while a site like this is very encouraging and we want to do everything we can to keep these trees healthy, it is not a high enough population of adelgids to decide to come out and release a whole bunch of biocontrol agents. We need a substantial population of the adelgids at any particular stand to release the beetles so that they can feed and reproduce and survive in a sustainable way. and the numbers of insects here aren't very sustainable. But on a positive note, the predators have been released in areas in this national forest miles away from here, and we found in recent years that the beetles themselves have spread on their own, and we find them in areas not anywhere close to where we release them. So it's quite possible that they're already in the site. There is ongoing silvicultural research that is examining whether opening up a stand and providing direct sunlight to the hemlock trees will result in reduced colonization and increased shoot growth. So it's too early to say and give specific prescriptions on how to approach this. Logic tells us that opening up the stand would be beneficial, but we're also cautious until we see the data and it really justifies that prescription. So currently what we're recommending to land managers is an integrated approach. If there are trees that, like these mother trees that they want to protect and they think they're at risk, to treat those, either soil injection or stem injection with chemical insecticide. That will keep the Adelgids off those trees, keep them reproducing, providing for regeneration in a site, and then releasing, subsequent to that inoculation with the insecticides, releasing subsequent to that the predators on sites to allow those beetle populations to build up and provide a sort of sustainable approach to managing this Adelgid pest. And then 3 to 5 years later, after the insecticide starts to wear off, then managers can decide whether to retreat those trees or to leave them alone and let the natural enemies do the rest of the job. And that is up to the land managers. So there really isn't an analog to this situation in Western North America, because the Adelgid is also native to Western North America, and it's on Western hemlock and mountain hemlock, and it does not impact those trees in a negative way. So it's not a problem, and there's no recommended management for the pest out there, for the insect out there. The reason we have the problems here is because the trees aren't adapted to the Adelgids and they decline quickly, and there are no natural enemies out here. So we're trying to provide short-term resistance with the insecticide treatment, follow that up with the release of the predators. If it turns out silviculture is a viable option to integrate into this two-pronged approach, then we'll have 3 things that we can do at a site.